I'm Steve Reed. I'm not from Harvard. I work at IDRI, which is probably a place in Seattle that most of you haven't heard of. And I'm going to talk about adjuvants, a little bit of jargon, but these are, as the Latin name would suggest, molecules and compounds that help the immune response. You've all been exposed to them, but you may not have known it. Just a little bit about the organization. IDRI is a nonprofit biotech. So we make products for neglected diseases. Yeah. Can you hear me okay? Yeah. So we're intentionally nonprofit. I'm sorry, Larry, you're right. And I really want to thank Larry for inviting me to here, and because I've been trying to get together with groups down here for a while to learn more about what's going on, and especially in the diagnostic area, to be able to translate this to the fields where we work, which is global health. We started in 1993. We've been around 20 years. We've spun out some biotechs to help us do what we needed to do. And one of the things we accomplished was making the first recombinant or defined TB vaccine. Now is in phase 3 by a big pharma company, GSK. But also we worked on adjuvants. I guess I don't need to stand there like a dummy. We work on adjuvants, which I'll talk about today, and actually helped develop, but frankly just bought a company that had already developed largely the only adjuvant that's in any approved vaccine by the FDA, which is a major milestone back in 2009. And this is Cervarix, the vaccine for HPV that GSK markets in many countries. We've also worked a lot on an intracellular organism called Leishmaniasis, which I'll talk about. But to do what we do and to make vaccines for diseases for which there are no vaccines, such as tuberculosis, leishmaniasis, we need a new category of adjuvants. We also need them for other reasons. We need them for responding to global emergencies to make your response more rapid and be able to use less vaccines so you'll be able to cover everyone. And as we grow older, it's evident that adjuvants can help address what I call immune senescence, because they can help stimulate the immune response as vaccines that people took 50 years ago or more wear off. So I'll talk a little bit about the adjuvants that we develop, but also more about the applications of these adjuvants. Background, well, you've been taking vaccines for a long time, and these vaccines have contained natural adjuvants. Adjuvants that we all use at IDRI and other places, the majority of these come from natural organisms, viruses or bacteria. And these are some of the milestones with developing vaccines along the way, but just what I pointed out is that it wasn't until the mid-2000s that modern adjuvants began to be included in vaccines, mostly by GSK. But nonetheless, many of these early stage vaccines had natural adjuvants, and as you know, many of them were then absorbed onto alum, which we know is an effective adjuvant, even if we don't understand it completely. It was never really approved. It was just grandfathered in. That's why I say that only until 2009 were adjuvants first approved, even though they've been in vaccines, for a long time. And these are the kind of molecules that I'm talking about up here, RNA, DNA, and lipoproteins. Things that the immune response itself doesn't respond directly to very much. You don't make a lot of response to RNA or DNA or lipids. So they can be put in together with a protein vaccine to help make that vaccine more potent. And that's what we really want to do. Not just to make it more potent, but I said to make it work at all. Because if you need a vaccine that stimulates T cells, you're going to need adjuvants. There are very few adjuvants in approved vaccines. Notice I don't say approved adjuvants because they're never approved by themselves, but in the context of a product. And they come into really only 3 categories. Alum, which again is in various childhood vaccines, it's grandfathered in, or oil and water emulsions. These are low amounts of oil, 2% generally, 4% in the case of the ASO3, in water, and these have been used in hundreds of millions of people in pandemic flu situations, rightly or wrongly, primarily in Europe. And then the last category of approved adjuvant type is a virusome or a liposome based, which is approved in Europe in flu and Hep B. So there's really only 3 platforms: alum, emulsions, and virusomes. And here we have that molecule that GSK approved that is used together with an alum to increase the immune response, so they say, to Cervarix, and where you can give a vaccine that contains relatively few serotypes but get a response that's much broader to multiple serotypes. And I happen to believe that's true based on our own information that I'll show you. Now, here's some jargon, but I'll get over it quickly. A TLR is a compound, a group that is responsible for stimulating the immune response. And what we've done at IDRI is develop the first series of synthetic compounds, whereas the first TLR compound used, and the only one in any approved vaccine, is a natural product that I'll show you in a minute. That was possible, developing these synthetic molecules was possible because of the crystal structure of a human molecule on immune cells that recognizes these structures. When we saw this crystal structure, we could define more carefully the composition, number of branches that you see here. These are lipid chains on a sugar backbone. The number and length that would fit ideally into this loop. And that enabled us to optimize vaccine adjuvants for people, not just mice, where most people do their work. And it also enabled us to get something much more pure. So this is monophospholipid A. I kind of grew up with this molecule. It was developed in Montana, where I went to graduate school. And it's a great adjuvant. It's the only one, as I mentioned, that's approved in any vaccine that stimulates through this TLR pathway. But it's a complex mess. You have to be a big pharma company to make this because it's a natural product. It's purified from a gram-negative organism, one of those red ones that we saw earlier. And you have to grow billions and billions of them and extract the lipid with solvent. And only 1/3 is really active in people. Whereas if we synthesize our molecule, we get 100% of this hexaform, 6 chains, and it's all active in people. What does that mean? It's more potent? Well, yes, but that means you can use less. We are a global health organization. You can't transfer the production of this anywhere you want to. This is made only in 2 places in the world, and it's made by Big Pharma. This I can synthesize anywhere. Right now it's being synthesized on 3 continents, and I can formulate it to make an adjuvant anywhere. So it'll fulfill our mission, which is global health. I'll skip over this. And this is an example. This is the natural product. This is the potency on human immune cells. This is one of the assays that we use to define the adjuvant potency. And here's just 2 versions of the synthetic. Here's the natural. As you can see, it's more potent, so you can use less. And the great thing for us, even though it's kind of a minor detail for this organization, for this group here, that this is the U.S. made, and this is one we just got made in India. So we can transfer the organic synthesis of this to India and have it made just as well as we can anywhere else. But an adjuvant molecule isn't an adjuvant. So what we do at IDRI is we formulate this into nanostructures. These are either made from oil or lipid in general. And these can all be filter sterilized, made in huge amounts, and again, made anywhere you want to. Now, you have to go there and help set that up. We just developed a formulation center in Pune, India, and we're working on one now in South Africa. But again, once you can make this molecule in big amounts, then you can go to these local sites and formulate it. The formulation that we like to use a lot are these emulsions that I talked about that have been used in hundreds of millions of people again, but they are not necessarily new. And I just love this little bit of history. As I was doing a review article last year or two, I came across something from the literature called Lippo Vaccines. And this was from France. And what they were doing is taking dried bacteria and mixing them with lipid. And why? Dose sparing. That means use less, so they can dry their bacteria and you get more doses out of one vial. And dosage sparing. That means 1 or 2 shots instead of 3 or 4. And who is interested in this? The military, to immunize soldiers against salmonella and other diseases. A lot of work was done in the U.S. At Rockefeller University and other places on these Lippo vaccines. And they led to what we started using is water and oil emulsions. That means a little bit of water but a lot of oil. Well, these hurt and they were largely dropped. But in UK, Salk's flu vaccine, influenza vaccine, was actually approved and given about 1 million doses. And they actually worked pretty well, but it hurts a little bit. So instead of a lot of oil and a little water, now we go to a little oil and a lot of water, and that's what's used today to carry our adjuvant molecules. So what I'm showing here is you can take a lot of different TLR stimulators and these are just a list of some of them, but you can take them and formulate them in different ways, so you can get quite a variety of adjuvants. And where this becomes interesting is not only for simple formulation for infectious disease, but then more complex formulations for cancer. And one of these right now is in late-stage clinical trials for cancer by one of the big pharma companies. But why else do we use adjuvants? Well, antibody diversity. Why is this important? You've all heard of the universal flu vaccine. Everybody wants to get a vaccine that will protect more than one season, or, as in last year, didn't even protect in that season very well. So we don't know when we isolate a flu virus every year if it's going to be the right one. So if you had an adjuvant that could help spread the response to a variety of different flu serotypes, you'd have a lot better chance of getting protection in any given season and maybe across seasons. And we published a study a few years ago showing that when we target these so-called toll-like receptor molecules on the human immune cells, we actually increase the diversity of antibodies made. Not just increase the amount, but the repertoire. And this is finally given us a way to understand how these things work. This is the number, for example, of antibody families that you get if you just give this so-called emulsion that I talked about. It's not so great. If you add our GLA, that synthetic molecule that I showed you, to this emulsion, you increase by over a factor of 10 the number of families of B cells related to the antigen that you gave, to the vaccine that you gave. How it translates in a real system is shown here in influenza. This is a common vaccine we take in the U.S., made by Sanofi Fluzone. This was a few years ago when we immunized animals with Fluzone. They don't respond too well because we only give one dose, and these are naive animals. But if we put emulsion in there, that oil and water emulsion, they respond better. If you add our GLA, it's a little better, but no big deal. This is the response against the strain they were immunized with, one of the strains they were immunized with. But if you actually look at strains that were not in the vaccine, how do they respond to those? These were strains from past or future years. There, the addition of this toll-like receptor agonist, which is so important, made a difference of life or death to these animals, whereas the emulsion alone could not do it in most cases. So this is broadening the immune response from a theoretical basis to a practical basis. And it's not just in mice. This is non-human primates as well, because a lot of these things can't be translated easily from mice to humans. But in this case, we see the same thing, whereas the Fluzone alone or with emulsion didn't do much to drifted antigens, where adding the GLA in these monkeys did a lot to increase the breadth. So this is a very interesting approach to broaden the immune response. And if any of you are looking at collaborations, and we've started to make these flu chips to really look at this in more detail, not only from seasonal, but from pandemic flu strains. And we've got thousands of time points now we can read on a chip with lots of different molecules that are the target for the antibody in flu. And by using this kind of informatics, we can test the ability of an adjuvant to broaden the immune response much better. So that's broadening, we call it, increasing the antibody diversity. Now we also want to increase the antibody response overall, and this is what you want when you need a rapid response. If the green cloud descends and you need to have a response right away, or if a new H7 virus comes out and we haven't got enough vaccine to treat everybody and we need to actually use very small amounts. I mean, I was at a WHO conference a few years ago, and the southern vaccine developers, represented by an individual from Brazil, said, we know that we're all going to die in the pandemic because there's 6 billion people and the capacity is only 1 billion doses of flu vaccine. So this is what we tried to address. And we did a clinical trial with a US company called Protein Sciences, where we immunized people with a vaccine that they produce in a baculovirus, a recombinant vaccine. But to get a barely approvable endpoint, which is this red line, it's 40% conversion of antibody, they had to use 130 micrograms. When we injected individuals, a human trial now, with our low amount of our adjuvant, only 1 microgram, we were able to show that 4 micrograms of protein, more than 30 times less, gave an even higher response than the unadjuvanted protein. So this is a simple way to address supply. And it caught the attention of the White House. In fact, Obama's asked us to do this. He had a meeting. He said, "Why are we still making flu vaccines in eggs?" And my response was, companies make a lot of money on flu vaccines and eggs. But when you start talking about new platforms, new ways to make vaccines in recombinant systems, defined systems, this is where the adjuvants come in. And I thought that it was pretty interesting that they paid attention, even if it is a form letter, I don't know. So this just came out. We presented this last month at a World Vaccine Congress in Washington. And this was very interesting. I want to talk about some of the places that these technologies are going. We did a study with a company called Medicago, which I like very much. They're in Montreal. They make vaccines in plants. And this was to answer a directive that we got, not just from the White House, but from the military that said, make us a vaccine that we can ship through the mail in the event of a pandemic. Something that can be self-administered. So we said, okay, we don't know if we're going to ever want to do that, but we'll certainly take the money and do it. We'll develop it. So we did that. And this was a trial that we did in collaboration with Medicago. And if you haven't looked at this technology, I'd advise you to do it because it's catching on. We're trying to help get this started in other places. But as you see here, you clone the genes into a virus that infects tobacco leaves, and you can start production very quickly. Of course, there's no animal products in this. You can scale it to massive amounts, and the product that you get out of there is very clean. It's called a virus-like particle, which is great because it resembles a virus, which is very immunogenic or reactive to the immune system. Whereas we just make a protein in any old synthetic system, it's often not very immunogenic, not very potent because it's not a particle. So these tobacco plants have figured out how to do this in a way that's very safe for us. It's not a virus you're immunizing people with. It's a protein that these viruses secrete. So we combined the vaccine antigen, the pandemic flu protein from this company, Medicago, with our adjuvant. And remember, we had to ship it through the mail, so we got this little needle from a company in Israel that I really like very much. And if you ever want to look at it, I'll be glad to show it to you. And even though it's a long ways away, even if you were close, you couldn't see it because it's microneedle. And these are 600 micron needles, in other words, very shallow. They can go right into the skin where the antigen, where the immune cells that you like the most are located. And it doesn't hurt. But more importantly, it works very well. And we did some studies early on to show that, again, with our adjuvant system, even if you immunize with the H5 VLP, you get responses to H2, H1, H16, so forth. So it does a great deal of response broadening. The seroconversion rate is here. We haven't finished analyzing the data yet. The bottom line is that you can give the adjuvant and the vaccine either muscular or intradermal, and you get good seroconversion rates. And this is using 20 micrograms of the vaccine, whereas the standard vaccine that's available, that was stockpiled by the Biodefense Agency, BARDA, you have to use 90 micrograms of it, and you only get 40% seroconversion rate. So this is another proof of dose sparing with another clinical candidate. But I'd talk about two more vaccine targets just for a few minutes, one of which is called Leishmania, which a lot of people haven't heard of. It wasn't on one of the big killer lists that was put up in the last talk, but it's a great model to teach us about how the immune system handles complex organisms. And things that we've learned from Leishmaniasis, from studying this organism, not only can help the patients, of which there are millions in the world, but they help us understand how to approach cancer, tuberculosis, etc. These organisms, you can't see it very well here, but this is an immune cell. This is one of your white blood cells that's supposed to go around and gobble up things and kill them. That's what they're paid to do. But in the case of Leishmania, they don't do it very well. These organisms just sit there very happily in a vacuole, multiplying, and they thumb their noses at these enzymes that your immune cell is supposed to be making. So we took an approach a long time ago to start identifying proteins of these organisms that can induce a potent immune response. And one of them, and I'll just talk about one protein, not all of them. We've cloned hundreds of them. But this particular enzyme is present not only in the parasite, but in yeast. And it turns out to be a target for amphotericin, which is a drug, an antibiotic we just heard about, a type of antibiotic that actually is very effective against both yeast and Leishmania. So, again, you learn a lot from studying these things. But this vaccine protein can be used to make a defined vaccine against Leishmaniasis. And what we do at IDRI, okay, so you have a protein. You want to make lots of it for the people in India, Brazil, Ethiopia, Sudan, where this organism lives, and in the Middle East, where it's huge in terms of cutaneous Leishmaniasis. You want to make a vaccine that protects millions and millions of people. But you don't want to make it necessarily out of just one protein. So we fuse several of them together and express them with a synthetic gene in a recombinant system so that one bacteria can make a vaccine protein that's really a multi-protein or a polyprotein. That makes your production costs much, much cheaper. So we use this SMT as a backbone and add antigens onto it. Now, that's part of the story. You have to have the right proteins. So as I said, we've screened through hundreds of them in animal models and using patient cells to test their responses over the years. But if you don't formulate it in the right way, all your work is gone and you'll never be able to evaluate a vaccine, because if you put it in the wrong adjuvant, the ones you can buy from your local Sigma, you won't get a response that's meaningful and you'll think you don't have the right protein. You have to have the right adjuvant because this response is dependent on T cells. And none of the vaccines that we use today, none of the ones you've taken in your lifetime, target so-called T cells. And you've read about T cells, so it's not jargon anymore. It's even in Time magazine. These are the cells that are responsible for killing viruses, killing tumors, and killing Leishmania. But if you formulate, this is our first generation clinical vaccine candidate, if you formulate an emulsion alone, you don't get very much of interferon, which we all know is a potent mediator of killing. But it's very active in terms of turning on another kind of immune response that you don't want. How do I know you don't want it? Because this formulation, SE alone, gave you no protection. These are parasites growing in a mouse foot pad, no protection. When you got interferon with these two adjuvants and no interleukin-5, you get protection. So this is very predictive of what you want to do for humans, and we make these formulations then for humans. The other unanticipated, and I say still not well understood, advantage of fusing antigens together is the spreading of immune response. If you formulate, this is one of the vaccine antigens, we call it Nucleoside Hydrolase, another enzyme, very interesting. If you immunize an animal with this, you get a response to it. If you immunize this same amount fused to something else, the response is even higher. If you immunize it with 3 proteins fused together, look at the response to that NH. The same amount of NH, but it has help from these other antigens. In other words, immune cells that are recognized in all these pitch in and amplify the response to any individual. That's another advantage of having a fusion. And it also more mimics the natural situation because the parasite doesn't consist of one protein, it consists of a lot of them. Your job is to figure out which ones will protect and which ones will make things worse, because some of them will make things worse. And the other reason we make fusions is because they work. These are protection experiments, again, where this is a visceral leishmaniasis that gets into the liver, another strain. And if you infect these organisms, don't treat, this is the number, this is the percentage that we assay against, 100%. All the individual proteins work to some degree, but when you start fusing them together, they work great. So this is what you would do to justify to a regulatory body making these fusion proteins. But I want to talk about adjuvants and their role in vaccine therapy, because this is another paradigm for testing vaccines, and not only that, for addressing things like drug resistance. I thought it was very interesting, the data that we just heard, even though they're anecdotal to some degree, 25 to 50% antibiotics aren't needed. I can tell you in Brazil, it's a lot more. They're so available, anytime you have a headache, you go get erythromycin. Drug resistance in these places is huge, and it's coming everywhere. You've read about it in the Wall Street Journal, the India issue in Mumbai with MDR, multi-drug resistant tuberculosis in the hospitals. As was just mentioned, people don't take the whole course. Ideal setting for drug resistance, tuberculosis. We get the same thing in Latin America. But let's get back to the vaccine part of it. There are strains and species of Leishmaniasis, this bacteria or this protozoa that I showed you, that can't be cured. This guy took treatment for 2 years. He lost a kidney because the drug that he took was antimony, which is nephrotoxic and cardiotoxic. Bad. They're desperate. So we started a long time ago treating these people with a combination of drug plus vaccine. And wow, even some of the early attempts with crude vaccines worked. So we evolved this to use a recombinant vaccine, one of these defined ones that I just showed you could protect, and published this paper not too long ago, where we could compare drug alone with drug plus vaccine. And what we really saw is a much more rapid cure with the combination of drug plus vaccine. The green line here is drug alone. Over a year, at the end of a year, you've got barely 50% of the people respond. Now, that's no way to live. If you have to keep taking this, it's very toxic, it's very painful, and these people have to work. I mean, if they're taking drug and all the time they feel sick, they can't produce for their families. If you add vaccine to that therapy, within 90 days you have 70%, within 150 days you have 100% people cured. A very innocuous 3 shots of vaccine during the course of antimony. Everybody gets the drug, but these guys got a placebo with the drug, these guys got a vaccine with the drug. Another way of looking at the data here. And another very interesting thing to think about if you're developing vaccines either for prevention or therapy, more is not better. And even though the numbers are small here, these data are true because we've expanded it into bigger studies. 5 micrograms of vaccine, 10 micrograms, 20. In tuberculosis and leishmaniasis and so on, there's always a middle ground here. Lower or too much is worse. So now we have a way, and we're working on getting this vaccine registered in Brazil, to treat people with a low dose of vaccine with drug and get 100% cure. I won't go into this, but I will. Those data that I just passed by are jargony data. But what they show is, which is counterintuitive, what kind of idiot would think that you can vaccinate somebody who's already got a big lesion like you just saw in that person's leg? They've got plenty of parasites. Their immune response is very vigorous. They've got a lot of antibodies. They've got a lot of T cells. But they're not doing their job. And you can understand this from the allergy field. Many of you probably were treated for allergy with desensitization. I don't know if you were or not, but I was. I was allergic to bee stings. Well, I had a huge response to bees. It could kill me. But what did the allergists do? They start giving you low doses of bee venom and building up gradually over a year or so until you actually change the kind of immune response you have. And that's what we're doing here. We're redirecting the immune response. We're redirecting it to the right antigens. So the vaccine contains the right proteins that are the targets, but it also contains the adjuvant that helps redirect the response. And the adjuvant that we developed at our first biotech is actually in the market in Europe for helping allergy treatment because it shifts the response away from inflammation to protection. So counterintuitively, you can vaccinate somebody who's already got a good immune response because it's not the right one. Let me talk just a little bit about visceral leishmaniasis to get people thinking about this because I need a lot of help here. This is what we work on in India, Bangladesh, Nepal. That's Asian version. And then in the Horn of Africa, Ethiopia and Sudan for the African version. They're slightly different. We don't understand how yet. The species are still called the same thing, but the diseases are different. This is carried by a fly, just like that other one I showed you. There was a little fly on that slide. I didn't point it out, but it's like a little mosquito. So these people are just out doing their business, trying to make a living, and they get bit. And they'll develop either those ugly lesions on their skin, which can be disfiguring, and they can even go to the mouth and nose like you saw in one gentleman, or in this form, the internal form, it's spleen, liver, weight loss, lymph nodes, death. And it's death due to the destruction of your immune system and your red blood cells and your platelets. So we need a vaccine for this because you're never going to eliminate all those little mosquitoes. And it's a bad disease. And one of the first things we did at IDRI was make a diagnosis because when I lived in Brazil, this kid had to undergo a bone marrow biopsy. It means sticking a dull needle into his bone marrow because there weren't very many of these needles around. and they often weren't done by trained individuals. And then look under a microscope for the parasite. And if you're lucky, in 50% of the cases, you'd find a parasite. And it was also shaped by my horrible experience at Cornell Medical School, where I was asked to come over and look at a slide from a little girl from South America. I and my chief, to see if this was leishmaniasis or not, so they could define her treatment. And it didn't look quite typical, but based on her history, we both agreed that's what it was. She was treated for Leishmania, and she died of fungus. So I never forgot that, and we started making diagnostic tests that were a hell of a lot more precise. And we developed a test that would diagnose visceral leishmaniasis and give the clinician an alternative to bone marrow biopsy. I'll tell you about those in just a second, but this is one of the sequelae of visceral leishmaniasis. It can get into the skin. Post-Calazar dermal leishmaniasis means after that internal form that those kids have, a lot of them will go on to develop the skin form. Not only ugly, it's full of parasites that the fly can pick up and transmit. So we developed a couple of forms of rapid diagnostic tests. The first one was FDA approved and adopted by India and Bangladesh more than 10 years ago. It's the #1 diagnostic test for visceral leishmaniasis in the world. It works great in Asia, but in Africa, it still wasn't perfect. So we developed the second generation and are just finishing the tests of this. And this is not a very good slide, but that's okay. One of my big problems is getting diagnostic manufacturers to take this market seriously, because it really isn't a market, especially in Africa. But I bribe them. You know, I say, okay, you can have license to our TB, or you can have license to our Chagas disease, which is present in the U.S. So, you know, okay. So they make these rapid diagnostic tests for us. But there's a new paradigm. This little girl here, she's not sick, but she's infected. So I need your help to figure out how to detect these individuals and proactively prevent them from getting sick, because once they're in disease, they may not be even treatable. And this is a study where this girl has a PCR. These kids have PCR positivity that drops over a year. Their antibodies stay high, but their PCR means they have DNA of the parasite in their blood. So we know that she's infected, but we wouldn't have known this until we developed new diagnostic tools. So I need help in getting the next generation diagnostics out there so we can proactively vaccinate people. I could give them drugs, except these drugs are too toxic, so I wouldn't do that. But if I could have a diagnostic to prevent infection, then I can give a vaccine to prevent infection. But also, this is very important, I want to leave you with this, the post-exposure prophylaxis setting to prevent disease. And lastly, we just published this paper on therapeutic immunization against tuberculosis, another situation in which you can use drug plus vaccine in an infectious disease setting to treat drug-resistant tuberculosis. And for those of us who need a boost later in life, we've also published that immune cells from old people recognize these adjuvants very well. So there's a new growing trend in industry to help use these adjuvants in immune senescence. I want to just thank all the people that have funded the development of these adjuvants and vaccines, and the people at IDRI. It looks a little bit like Colorado, but that's Mount Rainier. Thank you.
Thank you very much. I had a lung infection with hepatitis C virus, got over it with conventional therapy, and I was just wondering, doing research in molecular biology, epidemiology, and virology, as far as the antigens were concerned, would it be of any advantage to harvest blood from people that had been cured of hepatitis in general, since it is a pandemic, and using those antigens as a possible vaccine or modality to treat it? I just thought that somewhere in the genetic makeup of those individuals, there must be some trace of those antigens still available to harvest, and it would seem to be a great modality to me. But anyways, I was just wondering if you could explain what an antigen was.
Yeah,
Thank you.
No, that's a good question. That's kind of the approach that we used to make these vaccines. We used human cells and syrup to define these proteins. Antigens are proteins that are recognized by the immune system that are foreign. And yeah, it's a good approach. The Hep C vaccine field just took some stumbles. The models aren't very good. They used chimpanzees for a while. They had vaccines that actually made the disease worse. And so the movement's really gone toward therapy. But now there's some action on using a class of therapy that can stimulate the immune system as well as kill the virus.
I'd like to, I'm over here, Steve.
Okay.
For therapeutic immunization to help with drug resistance or drug-resistant bacterial infections, is the reason that I've not heard much about them, except the last slide you showed, is that the time course of most of these infections is too slow to think that there's an immunization step that could help with antibiotic treatment. Does that make sense?
Yeah, so it's about why haven't therapeutic approaches been used. One of the things that poisoned it is into the area of tuberculosis, the Koch phenomenon, where Robert Koch actually gave people BCG or killed mycobacteria, whole cells, and they died. It's a hypersensitivity disease of the lungs, and if you aren't careful, these things can stimulate inflammation. So it'd be like you trying to desensitize somebody with allergy by giving them a whole lot of allergen all at once, they're going to die. So it has to be done right. And TB was set back a long time, but complacency set in too. We had drugs that worked, and I think largely that's why. Now, there are situations where post-exposure has been done, rabies, for example. You want to, actually, tetanus, for example, you've already stepped on that nail, you have a little bit of toxin, you want to immunize quickly. But in large, therapeutic vaccination has been laughed at. Therapeutic vaccines have been thought of as a oxymoron, and we're still trying to get over that.
In the therapeutic vaccine trials, the drug that you used was not Ambisome. Did you try Ambisome with these things?
Yeah, therapeutic vaccine trials. So Ambisome is drug of choice when it's available for the visceral form of Leishmaniasis, frankly, mainly in Asia. It does nothing against these cutaneous forms of Leishmaniasis. And then this was a trial not only to speed the healing, but that guy that I showed you with the bad face, he started his problem with that single lesion on the leg. So they can evolve to the mucosal. So the other reason to be more aggressive in the treatment is to avoid mucosal disease, but Ambisome doesn't work. Nothing works, even Meltepicin, none of these things. You have to use antimony, unfortunately.
Yeah, I'd like to point out there's one area in which therapeutic vaccines have recently become very fashionable, and that is for cancer. That is, the individual often is already immune to the cancer but not doing a good job, and now you take all the brakes off the immune system in various ways, and in some cases successfully deal with the cancer.
Absolutely. While most of the drugs are antibodies or passive immunotherapy, I think some of the data coming out now, I'm talking about most of the ones that are approved, the antibodies, but you're right, now there aren't any active vaccines approved yet, but I think there will be. I think MAGE will probably be the first one next year where you're actually vaccinating somebody and developing an immune response. Actually, at Crixa, we had the first cancer vaccine ever approved for melanoma, and then the second one, you're right, it's the dendritic cell process from Bendrian was the second one. Yeah. Right. Absolutely. Antibodies.
Hi, could you maybe just comment briefly about adjuvant safety, side effects? I'm a veterinarian, and our profession is extremely concerned about sarcoma, vaccine adjuvant-induced sarcomas in cats. So we have roughly 90 million domestic pet cats, and it's such a significant problem that we're told where to vaccinate so we could amputate a limb if necessary. So can you talk about adjuvant safety?
I'm not sure what the adjuvant that is, but part of the reason that oil was lowered in some of these formulations was absolutely for that. Some of those people that were immunized in the UK in the 60s had persistent sterile abscesses or sterile granulomas for years. Look, adjuvant safety is complicated. You have to lower the risk. I think there's been a lot of issues with using too much because they haven't been formulated, either too much oil or too much TLR stimulant. And now we're understanding that too much is not enough, it's worse, and so we're getting much better. But now everybody's using synthetic oils, for example, and lipids to avoid those granules. I think 10 years from now, there won't be any oil vaccines anymore. It'll all be lipid-based. Yeah. Good, and I really apologize for speeding through it. I'm usually good at that, but I looked at my watch and figured it was 10:30, but it wasn't 10:30, was it? It was 10:15. So
We're back sharp at 11 o'clock, and in the meantime, Steve, thanks very much for a fantastic talk.